Good afternoon, everyone. Today, as we complete 10 years, we would like to give you a bird's eye view of how TIFR Hyderabad researchers came up with resourceful solutions in the wake of a global health crisis. Through sharing these inspiring stories from the lab bench, we would like to highlight how one tackled a scientific problem successfully taking it to completion. We also bring to you another recent development, the launch of our Science Media Center that will help us share more such inspiring stories in science. I would like to request Professor V. Chandrasekhar, our Center Director, to take us through the Institute's research goals and inaugurate our Science Media Center website.
Yeah, thank you, Anushila, and other colleagues from TIFR Hyderabad and other places. It's indeed a great pleasure for me to launch this Science Media Center of TIFR Hyderabad. And on this occasion, let me just tell you, although I will not steal MK's thunder, he's going to tell you about our history, how we started 10 years ago and came to this current state. But TIFR Hyderabad was formally launched 10 years ago, although we haven't really started operations 3 years since. That is in 2012 is when we started probably formally research activities. But even during these very early days, when we started activities at Narsingi in the rented accommodation, we were always conscious of the fact that TIFR Hyderabad had to reach out to society in terms of various activities. And so this got started in terms of popular lectures, and later somewhat of a formalized structure was there in, by having the Sawal Jawab at Lama Khan. I'm sure that more of this will become evident in our Science Media Center web page. So we were doing this both science as well as science outreach activities parallelly. And so this has now taken a shape where our Science Media Center webpage will put up not only our science highlights that our colleagues here and students have been carrying out, but also how we are interacting with the society around us. And I think it's quite nice that on this auspicious day of the 10th year of TIFR Hyderabad's starting, we're having this Media Center website being launched. Now, what have we been doing during these difficult months when COVID hit India? So we have tried from the very beginning, even during the very intense lockdown days, to see that we have a role to play in the country's fight against COVID. So we participated very actively in all the central activities launched by TIFR towards this effort. So we were part of activities like the COVID Gyan, which is the website devoted to giving accurate information about COVID. It's a multi-institutional website, and TIFR Hyderabad continues to be an active part of this, contributing both to its editorial and to its regular content. But we've also been actively participating in research of various fronts. One is relating to diagnostics. Can we come up with better, cheaper, and quicker methods of detecting COVID? And in this, we have been to a large extent successful in collaboration with other institutions in Hyderabad, the Nizam's Institute of Medical Sciences and the ESI Hospital in Hyderabad. Manish Jaiswal and his students and colleagues have come up with a new rapid diagnostic tests based on the LAMP technique. And this is a method that is now hopefully will become available to the public in the coming months. So this is one important development in terms of diagnostics. We've also been working on therapeutics, and this is of course a long-term sort of a research that is happening. And many colleagues have been working on this based on different approaches. And we have also, this is on collaboration with colleagues from both Kolaba as well as Indian Institute of Science.
There is a slight issue with the audio.
Can you, is it clearer now?
Yeah, now we can hear you. There was this little issue before.
Okay.
It's fine now.
So, yeah. So as I was mentioning, in addition to diagnostics, where we have had this LAMP-based technique now nearly ready for being taken up by a startup or by a company, we also been working on therapeutics in collaboration with other research institutes, including Indian Institute of Science, Bangalore. And also we've been working on other things like, for example, better masks. I think we'll hear today in today's talk something about this based on, and this was development based on graphene oxide based materials, and also better sensors to figure out if N95 masks, for example, are still functional, or have they reached a stage where they need to be discarded. And indeed, such machines or such instruments can be utilized for other purposes also. And an ab initio sort of a technique in terms of a brand new methodology of an RT-PCR, which is going to be much cheaper than what is existing currently. And so that is in terms of this, again, we'll hear today in today's talk. In addition, another important initiative of TIFR Hyderabad has been to see if we can assist to capacity building. So towards this, first we did a dry run in terms of training our own students and postdocs on RT-PCR techniques. And later we wrote up a research project to the Melinda and Bill Gates Foundation. And this has been funded through the PSA's office. And the formal launch of such a training program was done by PSA on October the 2nd. And we hope in the coming months to be able to actually set up one such center in remote parts of Telangana. So this is one of the things that might happen. As I was talking to Manish today morning, he was mentioning that this is one of the goals to be able to set up something in the hinterland of Telangana. If we can do that, that will be really successful. So I think with this, I would stop. So this is what we have been carrying out on COVID-19 in spite of all the lockdowns and all the hardships that we have to face. We did not stop our research, continued our activities. And I think to some extent have been able to participate in this national research activity on COVID. So once again, I formally launched this Science Media Center of TIFR Hyderabad, and it looks wonderful. And I hope that we'll have a very, very rich content which will showcase what we have been doing both in the science matters as well as how we are outreaching with the society. Thanks, Anushila.
Thank you, Professor Chandrasekhar, for providing this comprehensive overview of the Institute's COVID-19 related research activities and inaugurating the Science Media Center website. Now, I'd like to request Professor M. Krishnamurthy, who's a faculty at TIFR Hyderabad, to share with us how the Institute developed over the past decade and is set to grow in the upcoming years.
Thank you, Anushila, and congratulations for putting up with a very nice website, which is going to be very informative, I'm sure, and will actually guide everybody, every one of us, how to communicate whatever science we do and how to make it palatable to the larger community. I thank also Professor Chandrasekhar for giving me this opportunity. I can't believe that it's already 10 years since we have started in TIFR Hyderabad, and this is a journey that is exciting, suspense thriller, I would say. And I will give my own recollection of this. I'll share a presentation, just showing some bits of it and in some chronological order of things that happened. Anushila, you can see the screen?
Yes.
Yeah. So TIFR 2.0 or TIFR Hyderabad, as we call it, started quite some time back, actually. In 2008, the first germination of the seeds for starting a new campus started. We started looking for land. Initial survey, of course, was in Mumbai. We went to places like Taloja, Panvel, looking for a suitable area where we can start scientific activity and watching tour of TIFR. And at the end of some struggle, we came across the plans of the government of Andhra Pradesh, which wanted to build a scientific base along with the financial district that they were planning. And 12th July 2008 was actually when we first landed in Hyderabad looking for the land. This was the enthusiastic person who was showing us from a high vantage point of how the land was there around, and it's very, very nice land, as you can think and imagine. So further to that, very quickly, we wrote a proposal to the government of Andhra Pradesh requesting for the location of land. By November that year, a MOU was signed and 209 acres of land was given to us. Of course, it takes a long time to plan, to execute a big campus like this. So the initial ideas of how we project a new campus, or to identify the areas of interest, the cross-pollination points was, and this I would say is probably the 0th version of the life sciences and matter. Different people with different ideas came together, and bits and pieces of the research that they would like to do got started putting in place. And with that, we made a concept paper. It took us about a year or more. And in July that year, we presented a concept paper of TIFR-H to the Atomic Energy Commission. And very quickly after that, the AEC had Atomic Energy Commission approved this plan. And in October 2010, today, 10 years ago, Prime Minister unveiled the foundation stone. Just maybe for a minute, I can show you can you see this, Anushita? Okay, we had our invitation for this planned. Then further this, of course, after this event, of course, we had to continue a brainstorming session to divide the activity into a suitable, palatable pieces of convertible documents through which we can seek funding from the government and execute our plans. So November 2010, we had a brainstorming session with the then Atomic Energy Commission Chairman, Professor C.N.R. Rao, and we came up with a phase 0 plan by the end of 2010 and requesting a funding of about 75 crores from the DAE to start this activity. So this is what we called as phase 0. And even before the buildings were built, we very quickly started the activity, keeping up with the tradition of TIFR. We rented a building in 2010, which was supposed to be a marriage hall, converted it to a research building. And by 2012, enough people accumulated, graduate school started, students started doing their doctoral degree work in this place. And simultaneously, of course, we were planning for the first research building of our own in the campus. It again took some time. The planning and execution of these tenders by July 2012, in about 1.5 years from that point, we actually started the work, and it was supposed to be completed by December 2015. It took a couple of more years, and this is what we saw. I don't know if you can see this walkthrough.
You have to share your screen.
So we had plans pretty much worked out and we saw this virtually for a while. And over the time, of course, this became a reality. And as you can see, by 2018, we actually moved to our own campus, relocated all the 15 to 20 laboratories that were fully set up, and completed this arduous task actually in a year. Really thanks to the enthusiasm of all the people who were in the part of this business. And the new phase actually have started about a year ago. The detailed project report for the phase 1 of the campus development was approved by the Atomic Energy. The idea was to develop for new facilities for 40 more new faculty members, so that we take our total strength to about 60. In addition to about 150, 200 students that we have, we will add another 300 more, so that it becomes 400 to 500 students, and of course expand to newer areas than what we have already initiated. So we have started the architectural work with the vision of getting a 20-year master plan and begin the first phase of construction and hopefully conclude the first phase by March 2014. The motto is, of course, to have an architecture and design that promotes creativity and interaction in harmony with the natural landscape, responsible management of energy, water, and waste. We plan to build a 2 megawatt plant. So in the phase one, we will develop about 50% of the land. What it means is that 50% of the land will be more or less left untouched. Of course, we will be making it integrated campus with the land development, but keeping pockets of land that will be open for future modifications and building. We will have 4 new blocks. Of course, these are not going to be like the physics chemistry blocks of a typical university. This is going to be thematic blocks, depending on the kind of research related to life, matter, and light, and the crossroads in between. We'll have thematic facilities which are emphasizing on a particular discipline or a particular area of activity, like petawatt, NMR, imaging, high computing, HPCs, high power computing. We'll have common facilities like workshops, lecture halls, and so on. Other required civil structures. We'll also build guest houses, hostels, staff house, and everything. So we will broadly divide the thing into academic zone, residential zone, support zone, utility zone, and of course the natural zone, so that it gives us a room for a place for creativity and so on. So, and if you have not privy to this where we are located right at the back of Hyderabad Central University and next to the financial district. The big chunk of land comes in 2 pieces of roughly around 150 and 60. And of course, the third piece of land, which we have already developed, and it's not shown here. So this would be TIFR Hyderabad integrating with the financial district and the industrial development and the other kinds of developments that are happening in the geography, like the National Institute of Animal Biotechnology, the state-of-the-art sport facilities, incubation centers for taking research to the real world and so on. So I thank you for giving me once again the opportunity to present what the journey that we have gone through, and we plan to go through over the next 10 years or so. Thank you.
Thank you, Professor Krishnamurthy, for sharing the vision of the Institute. So at this point, I'd like to start a new segment of this program where we present the efforts undertaken by the Institute in response to the pandemic. When COVID-19 spread across the globe, it did get many scientists thinking about what they can do to help arrest its spread. And researchers in TIFR Hyderabad were no exception. They immediately immersed themselves in strategizing how one could take steps to combat the spread. This included efforts to develop diagnostics, organizing a COVID-19 diagnostic training program with a view to increase the testing capacity of Telangana, developing rechargeable N95 masks and testing devices, engaging in long-term research to devise strategies. Among these, we present to you 3 research stories, taking on the entire journey, right from when inspiration struck to the moment it worked. Before we begin with the talks, you're requested to write your questions on the chat box on YouTube. And the first story of the day is that from Sunana Sikar. She's a graduate student specializing in biological sciences. Today, she's going to talk about how she and her colleagues standardized the LAMP assay, resulting in a quicker and reliable COVID-19 diagnostic test.
Good afternoon, everyone. Thank you, Anushila, for this introduction. Today in this talk, I will discuss about our efforts in optimization of an alternative and faster method of diagnosis of COVID-19. The world seems to have changed all of a sudden since the first man was infected with the SARS-CoV-2 virus in Wuhan, China in December 2019. an infection that became a global pandemic in no time. Social distancing and wearing masks have become the new normal for us, right? And I must repeat that this is mandate, since we do not know who among us are actually infected by the virus. Frontline workers like doctors, nurses, path lab technicians, and also researchers are working day in, day out trying to find a proper cure, proper diagnosis, and treatment for these infections. And most important thing right now is testing. Testing, and more testing to detect those who are infected, followed by their immediate isolation to check the spread of the virus, thereafter, their proper treatment. And as you can see from these numbers here, laboratories across the country are testing so many patient samples by the approved method of testing, which is right now, that is RNA isolation and RT-PCR. So let me just talk about the procedure. Nasal swabs are being taken from patients and they are transported in a special media that can harbor the RNA virus. This COVID virus is an RNA virus, meaning the genomic material is RNA. So this viral RNA is then extracted and converted to DNA by mixing it with specific reagents and enzymes. And this DNA that you get is further amplified by something called the polymerase gel reaction, which is RT-PCR. So how does this amplification occur? DNA strands cannot be amplified by the enzymes just like that. These strands have to open up. And that is why RT-PCR comes up with this sophisticated instrument that, that is basically controlling the temperature regulation. So in this, we can run thermal cycles. At first, 95 degrees Celsius is used to separate the strands, followed by the binding of short stretch of nucleotide that will target your gene of interest and help in the amplification. These are called primers. These binding of primers happen at a temperature of around 45 to 68 degrees Celsius, followed by the elongation and amplification at 72 degrees Celsius. So from 1, you can get 2, and from these 2, this thermal cycle can be repeated to get an exponential amplification in the DNA count. This sophisticated machine here on the monitor then displays a complicated graph like this, which needs proper understanding. Essentially what it does, it tells you how much DNA starting material was there. So RT-PCR, being a gold standard of testing, can help doctors not only say whether a person is infected, but also it can predict how severe the infection is. RT-PCR, yes, it is a gold standard, but it definitely has its drawbacks. The expensive instrumentation, because the thermocyclers are very expensive. One has to have a proper skilled personnel to handle these instruments and to figure out the curves that are built alongside it. And starting from RNA extraction to the PCR, a successful analysis takes around 4 hours of the time. So essentially, we here at TIFR Hyderabad wanted to find out a better alternative. What we wanted was expensive instruments, go away from the door. We need handy and easy instruments. We need a simple procedure so that any unskilled or non-expert can handle this training. And if we can generate or develop or modify a method that can show results in 30 to 45 minutes, that would be best, because the massive Indian population needs to get tested at the earliest. This is when we came across a technique which was developed in 2000 by Sugunori Notomi called LAMP. LAMP stands for Loop Mediated Isothermal Amplification. So how is it different from PCR? How is it different is that the separation of the two strands here is based on an enzyme and not on temperature. So this enzyme can work at 65 degrees Celsius and all the entire process can be maintained isothermally, that is at a constant temperature. So the need for a thermal cycler is no more. And essentially, LAMP does not use 2 primers like the PCR. In LAMP, we use 6 set of primers. One set runs along the length of the DNA strand just like PCR. But the interesting thing comes with the second set of primers. If you look closely, you will see that a part of this primer is exactly complementary to the DNA strand here. So, when this primer is allowed to be polymerized by the enzyme, just like the one in the sister strand, what we get is that two complementary opposite strands fold upon each other, finding the pair and exposing a single stranded loop. A second set of primers that can modify and polymerize this loop is again used. So the entire DNA is being amplified and polymerized from 6 different places at the same time. Thus, you reach the limit of detection, the exponential amplification, in within minutes, like in 10 minutes or so. Thus, your time reduces and you can only need 30 minutes instead of 4 hours to get your result. And how will you predict the result? This amplification of the DNA causes a lot of proton release in the media, which will turn the medium acidic. So you can use pH indicators, which will just show the change of color. In LAMP, what we do is that we mix the viral RNA with the reagents and we do nothing. We incubate for 30 minutes at 65 degrees Celsius, and within this one tube, from RNA, DNA is synthesized. From DNA, the amplification occurs in loops, pH changes, and the pH indicator changes its color from pink to yellow, and we can see the color change. So we don't need an expert to analyze complex graphs, but anyone can see the change in color and tell you whether your sample is positive or negative. This method was developed by Nathan Tanner of New England Biolabs, and this method of LAMP has been in use even during the Zika virus outbreak, and people in Wuhan, China last year also tried to employ LAMP for COVID testing. And in here, in TIFR Hyderabad, we wanted to modify further on this technique and present it for the people, for the common mass living in remote areas who don't have the access to an RT-PCR. So in the labs here, we synthesized synthetic DNA plasmid and carried on this LAMP assay. And as you can see from this, with our negative, we have a pink color, and wherever we had the DNA, it changed its color in 30 minutes. But DNA is not enough, right? COVID is an RNA virus. So in the lab, we synthesized in vitro RNA from the DNA. We transcribed the RNA and we tested the sensitivity of this LAMP assay. And if I run you through this slide, you will see here the number of RNA molecules that we added was 0. So at 0 minutes and at 30 minutes, it stays pink. And this was RNA from a gene N, which is the target gene for COVID nowadays. We then did a titer and we can see that only with even 300 copies of RNA molecules, we can see a proper, efficient and significant change in color. That means LAMP is working. But this is not enough. If you get to a patient, you will not have such pure RNA. In the patient samples, there will be human cells, human RNA, and we wanted to mimic it in the lab. What we went on to do was take a human cell line called HeLa cell line, spike it with the synthetic RNA that we prepared, and exactly mimic the protocol that is being followed in the hospitals and do the RNA extraction. This RNA was then mixed with the LAMP reagents and the primers that were designed in Hyderabad. And we found even with that, the LAMP assay was sensitive till theoretical 300 RNA molecules. In practice, it should be less than 300 for sure. And after this, we sent it to ESIC and NIMS hospitals where our collaborators under Dr. Madhu Mohan They tested the same with human patient samples. We had given them synthetic COVID RNA that we synthesized, which they used for their positive control, and you can see that it turned yellow, which was expected. For the negative control and the patient samples, here they did not add any sample, so it stayed pink. And these three samples were already tested positive by the approved RT-PCR method. And you can see here also they are changing color to yellow. That means LAMP assay is corroborating its results perfectly and efficiently with RT-PCR. What we are trying to do now is develop further on this method, trying to see if we can get rid of RT-PCR and RNA isolation altogether. We are trying to do LAMP with saliva and nasal swabs directly. If at all we can do this, we are sure that LAMP can be at least almost as sensitive as an RT-PCR. Even though it is qualitative, it can reach to the remote places as Dr. Chandrasekhar was talking about, and we can get the massive population of India tested and properly treated, something which is at a grave threat right now. With this, I would like to acknowledge my teammates and our collaborators and the funding sources. I would like to take questions now. Thank you.
Thank you, Sunena. We have a couple of questions on YouTube. The first is, what do you mean by spike it with RNA when you mentioned this in your talk?
Okay. Hi. Thank you for the question. Spike it with RNA means when we take the cells and do the RNA extraction, we need to break the cell membrane. That is essentially called lysis. During this lysis, we add our synthetic RNA into that lysate. So now what you have is that you have RNA of the human cell line plus the synthetic RNA together, from which we carry out the RNA extraction procedure.
Okay, so we have another question here. This question is about, now that we have a test developed, what is the next step that you're considering to implement this in a large scale?
Okay, these tests are awaiting approval, and in a large scale, what we are planning to do is trying to get rid of RNA extraction altogether, like I said, because if you are taking nasal swabs or saliva samples, you always waste another 1 hour, not waste actually, but utilize another 1 hour for RNA extraction. What we are trying to do is, say, send up motor testing centers, which will have these reagents. We can take the patient sample directly and run this test. So essentially, mobile testing can be achieved if we want with this method.
Okay. So thank you so much, Sunana, for this talk. And now we would like to move on to the next talk. So the second story is about Parshvanath. He is a graduate student in physics. Parshva volunteered for the diagnostic training program in the institute. And during the program, he got to know about how a PCR machine works and started wondering why it was so expensive. In the middle of the pandemic, it would be crucial to make sure that enough machines were available for testing. So he decided to piece together a fluorescence detector. Today, Parsha is going to talk about how he made a toy version first and then a more upgraded, fully automated open source fluorescence detector. Parsha, would you like to share your screen? Yeah.
So am I audible?
Yes.
Okay, so shall I start?
Yes, please.
Okay, fine. So now I'll actually tell you or share you a story of piecing together a fluorescence detector. It's my own story, own favorite story, basically. So if I ask you to actually describe a scientist at work, what will you have in your own mind? You may think of something like this if you are eagerly awaiting for the COVID vaccine, or you may see something like this if you are a Star Wars fan. Anyway, of course you are right. These are the experimental branch of actually science where you observe or measure or test your own hypothesis. But there is another way to do. You deal with equations and graphs which is called a theoretical branch of science, where you develop models, you make hypotheses and make predictions. There's another emerging branch also where you write your computer code, you simulate your models, test hypotheses, etc. So now, this is the full circle of science. As a graduate student here at TIFR Hyderabad, I do theoretical and computational sciences. Now, but anyway, who does not want to complete the circle of science? So, just a minute. Yeah, fine. So, but when I was a high school kid, I always loved homebrew scientific experiments, electronics, and other do-it-yourself stuffs, which I was actually missing here. Then this happened. So back in May 2020, I signed up here at TIFR Hyderabad for the COVID-19 sample training program, diagnostics and training program. And of course, this is the first ever time I entered into a proper research grade biological lab. And these sessions used to be hosted by Deepa and Gopalakrishna. And I asked many questions to them. Well, so I asked them, I heard that there is an expensive instrument called the real-time quantitative PCR or qPCR, which is being used for the COVID testing. I said, I also heard that there's a scarcity of this instrument and there are no local manufacturers. Well, they said all are true, and they said it costs around 20 to 30 lakhs. Well, I said, well, that's a lot. But then I asked, how do they work? Then I came to know and understand. I'm just going to explain whatever I understood from them. So say for example, you take a vial, it's a container with water and shine some blue light, and you'll see the same blue light back emitting from the water. It's not that interesting, but now if you add some special particles or some molecules in that water and shine that same blue light, now along with the same blue light, you may see some amount of green light being emitted. Well, this phenomena is called fluorescence, and the special particle or the molecule, chemical X, which I've written here, is called a dye or a fluorophore. Well, so it turns out that this is how you get tested for a viral presence. So what you do is, it's exactly what Shunana has explained. You collect swab samples and very carefully bring it to the lab, and again you process it and you do something, then you get a final solution. Now you put that final solution in the small files which are shown here and load them to the qPCR device. And this is exactly what the qPCR actually does. So the heating block, so this vials actually sits on a heating block, and the heating block is periodically heated and cooled down, and it follows a particular pattern or a cycle, which is called a thermal cycling. And after each cycle, you turn on this blue light and see if there is a green light being emitted. So the thing is, after a certain number of cycles, it's typically 15 to 20, I think, if the green light shines, see, here is a green light. If it shines, that means that it's a positive sample. All right. So what's the big deal, and why such a device is so expensive? I became more and more curious to explore. This is what I found out. Well, you again take that pure water-filled vial and shine in some blue light. You get, of course, the same old blue light back and no green light. Now imagine this following scenario. You add 100 molecules in that pure water and you see some green light along with that blue light, which you see here, small amount of actually green light. You add another 100 molecules, so together you have 200 molecules. The intensity of your green light is precisely double now. Now, there's a nice property of this phenomena called fluorescence. The intensity of this green light, it systematically increases with increasing number of molecules. That number of molecules sort of in that solution, you sort of say the strength of the solution. And also note that the blue light is much more intense and it even persists without the molecules, without those special fluorophores. Now there is a problem. In a typical PCR case, the strength of the solution is so low that at the worst case, the intensity of the green light could be 10 to the power 9, that is 1 followed by 9 zeros times weaker than a typical room light where we are sitting now. So you cannot really see with your eyes or a camera. This I thought, but I was wrong. Well, but there is a solution. What I did is the following. I used a photodiode to convert that light to electrical signals. So again, there is a problem. So it's problem number 2. The photodiodes are extremely sensitive, but they cannot distinguish between colors, unlike our eyes. Now, for example, take the following scenario that, say, for example, you have a negative sample which emits blue light. And say this is a positive sample which is emitting blue and also green light. You may see that the photodiodes are giving you the same electrical signal as output. But so you basically would say that both samples are actually positive. But so that's the case. Now what you do, you use a proper color filter to block out that blue light. So here is that color filter. You can see now it actually blocks out the blue light, and now the photodiode only sees the green light. This is exactly how it works. And then what happens is, now the photodiode has converted that tiny amount of green light into a very small or tiny amount of electrical signals. Now I used electronics, I built those amplifiers, homemade amplifiers, to actually amplify those tiny amount of signals. And then finally we need a brain. That brain is called a microcontroller. Again, it needs quite sophisticated actually programming. I use that experience there. And finally a computer for other analysis. But I want to tell you that building this kind of amplifiers which would amplify such tiny signals are not easy. It requires multiple levels of actually filtering, and iterative improvements made this possible. Now I'll show you the first version. During the lockdown time, I did not have access to many components. So whatever I had, this Lego stuffs, this Lego building blocks, I made the first fluorescence detector with this, Lego blocks. So where you can see this solution is in the vial. This vial you can see here, that blue LED is here, which is actually emitting the blue light. This is the optical filter, glass filter. This is a photodiode and all other electronics. So these are the amplifiers, the brain, and a computer. Now I'll show you how I did that experiment. So these are typical PCR vials, and these vials actually contain solutions of different strength. In this case, left to right, it's 800, 600, 500. If you care about the unit, it's in nanomolar, okay? And then finally, I have pure water. Well, so there is an experiment. I have one detector, and these 8 PCR tube strips are placed here. Then I manually actually slide it, and then I'll detect the fluorescence. And this is the first result I got. Well, the proof of concept is established, but you see, it was supposed to be a perfect straight line. It's not a perfect straight line, but the trend is clear. Now, well, no experimentalist likes this scatter. So I tried to improve it, and there's a final version. So this time I improved this optical setup by using a combination of a special mirror and some other optical filters. And the amplifiers, these homemade amplifiers, are significantly improved. And the microcontroller, the brain, basically remains the same. Again, I took the same vial, filled it up with even weaker or less stronger solutions. So it's 256, 120, 864, and so on, all the way down up to 4 nanomolar. So here is the experiment. So I'll just a small video of this experiment, which I want to show you. I can't zoom it in. Oops. Oh, sorry. Just give me. That's a full screen option. Yeah. Just give me a second. Sorry, I can't see the full screen thing here. Well, you may have to see the, okay, I got it. Well, fine. So anyway, you can see this slider here. The PCR vials are sitting here, and it is being slided through this. And this is the intensity of the fluorescence. You can see that as the solution, as the strength is decreasing, the fluorescence peaks are also being decreased. And anyway, I can now speed it up. So here is the final result. Here we have the final result. Scatter is gone. This is a perfect straight line. This is the final result of the thing, which makes me actually happy. So now, here's the conclusion. So as I told you earlier, this RT-PCR is basically a combination of a thermal cycler and a fluorescence detector. The heating block or the thermal cycler, an open source design, actually existed, and it cost around 40,000 rupees. But for the fluorescence detector, which is a very sensitive light detector, that open source design was not existing. So now it exists, and it cost around 60,000. Well, now I want to just tell you before ending that why these open source designs are so important. In open source designs, you design it and you make it freely available to the actually public. And all of your designs uses mostly off-the-shelf components, which you can which you can buy from normal stores without going to any sophisticated things. And of course, there are no patenting or licensing or licensing restrictions, all this. And you can build it, use it for free. And if you're enthusiastic enough, you can come and improve the design. You study it and learn it and make science and knowledge open and free to the humankind. Fine. Now the acknowledgement. This is my team. Our team comprises of myself and Raja Lakshmi, Raji, and Pranav. Well, as I've told you earlier, that I'm from a completely different background. I do theoretical condensed matter physics here, and these are the real experts of this field. I received many helps and supports from them. And for other helps, I really want to acknowledge Shubhrojyoti and Shamosri, those who made those solutions, because preparing solution of that small strain, that weak solutions are really actually challenging. And also I want to thank Rakesh and Brahma, who made many small components from our workshop. They're from TIFR Hyderabad workshop. And lastly, I want to also thank my own thesis advisor, Surajit Sengupta, who actually let me go free and do all this. Finally, I want to thank Anushila also to actually help me to put together this story. Thank you.
Thank you, Parshva, for this really illuminating talk. And shall we take some questions now? I'll just see if we have any questions.
Yes.
Can you discuss your software? This is a question on YouTube.
Yes, I can discuss my software. The software is inside of this so-called microcontroller or the brain. Also, I did not put actually details of it because it's too much technical. Thing is, at the microcontroller, I am doing actually phase sensitive detection, which a typical lock-in amplifier actually does, but I'm doing it completely in software. So if you want to know, this microcontroller is a highly powerful, a microcontroller development board. This is called TINZ 4.1. So this TINZ 4.1 is really a powerful microcontroller board. It actually equips a ARM Cortex M7 architecture, actually microcontroller. So I was able to do all this modulation and demodulation things. These are at the heart of this PSD or phase sensitive detection in the software only. So this is a software which does this phase sensitive detection and some other stuffs, like it controls the movement of this PCR vial tubes, which is it does in brief. Yes.
Okay, all right. Thank you so much, Parshva, for this talk.
Yeah.
We would move on to the third talk of the day now. So Dr. G. Rajalakshmi and Professor T.N. Narayanan led a research effort where they came up with an N95 mask which could be rechargeable. And this third story of the day is about how they got inspiration, decided to work on the problem, and developed a very cost-effective and easily rechargeable N95 mask.
Hi, Anshila, can you hear me?
Yes, I can hear you. Please go ahead.
And you can see my slide too, right?
Yes.
Right. Can I start?
Yes, please.
Okay, thank you, Anshila. And good afternoon, all. So both the previous speakers discussed about how do we test whether we are affected or not. So I'll be discussing how to protect ourselves from infection. So we all know that, the COVID-19 pandemic has necessitated the proper use of the right respiratory mask. So World Health Organization has recommended this, and many countries, including India, mandate individuals to wear masks in public. But there are certain concerns with this existing so-called right mask while using them for a longer or extended period. I will define what I meant by the right one. So here at TIFR Hyderabad, with the help of these researchers, we have tried to address some of these existing issues by developing a novel respirator mask of high quality. I will also define what I meant by the quality of a mask. There are certain parameters. And the interesting aspect of this novel mask is viable for mass production. And the second part is at the how to test the efficiency of a mask. That's also important to know whether the mask is good for use in this particular condition. And after several uses, whether it is still efficient as good as it is in the initial use or not. But the mask testing equipment so far, to our knowledge, are imported, highly expensive. So the important point here is that, we understood the parameters need to be detected, and by finding the right sensors, we could develop a cost-effective, simple mask testing device at TIFR Hyderabad, and we'll be discussing some of these aspects too. So when I talk about the mask, what a mask does actually? Mask is basically a particle filter. The breathing air has different types of components or constituents called particles, can be combination of aerosols, droplets, dust, etc. And the important thing is that the bacteria and virus can be, within these kinds of aerosol droplets. So it has to be filtered. But the challenge in this filtration is that their size range in a very wide range. For example, the typical sizes is of the order of few micrometer. A micrometer is millionth of a meter. For example, hair is 100 micrometer. So, and it can vary, size of these particles can vary from few micrometers to sub-micrometer. For example, aerosols have the size of typical 5 micrometer or higher, or 5 micrometer or lower, or droplets can be more than 5 micrometer. So you need to have a very compact and efficient filtration mechanisms to filter out all these micrometer and sub-micrometer particles. So if you look into the SARS-CoV-2, the virus of today's concern, it has a size of around 0.120 micrometer. So it's a sub-micrometer, less than 1 micrometer. But when it comes within the aerosol, it has a size around 0.3 to 0.5 micrometer. It can vary. But so the particles need to be filtered is of this size 0.3 micrometer. There are certain standards for masks in different countries, have different names. So the very popular name what we heard these days is N95. Meaning of N95 is N95 has 95% efficient in filtering out the particles of 0.3 micrometer or larger. So this is a US standard, but similar standards are available in different countries and with the different names and grades. For example, FFP2 in Europe, KN95 in China, P2 in Australia, etc. There are various other masks are also available. For example, medical face masks, silk woven masks, cotton woven. So various kind of clothings can also be made into the mask. And they are all good actually. But if you test them in a similar standard for 0.3 micrometer, one of the reports says that the efficiency is a bit lower to the N95 standard. So these masks will work efficiently only when if you ensure the social distancing. So particularly when crowded places, you need to have a very high filtration. You need to have a mask of standard of N95. So how an N95 works? So as I told you, it has multiple filtration mechanisms. So all masks work on the first thing, the inertial impaction, where the non-woven fibers can filter out more than a micrometer very successfully using the phenomenon called inertial impaction. With the inertia of the incoming particles, when they see an obstacle, they can get stuck and they can be filtered out. But submicron particles can be still diffused into and inside the mask, and then hence it can be still cannot be filtered out using this kind of inertial impaction principle. But fortunately, virus-laden aerosols are charged, and hence a charge-based filtration may work. Fortunately, by the virtue of the synthesis method, these non-woven fibers in N95 have static charge. They got this static charge via the filter, the way they synthesize a melt blown or electrospinning method. But the problem here is that these fibers lose their electrostatic property in a day or so. This is well documented and we also study. So an N95 mask may not be effective when you are exposed to the ambient condition for a longer use. Hence, what is the solution to that? When we thought about this, the immediate idea what we come to our mind is, can we do a simple charging via rubbing? Because this is well known to us as tribo charging. If you use certain combs and in certain conditions of hair, you can see that the hair gets charged. What is happening is that a static charge separation is being generated between the comb and the hair. And how much DC voltage is generated in between depends on the materials you are used and the hair condition for sure. So if you look into different materials have different tribo charging capability. Some of them get positive charge and some of them get negative charge. So if you take candidates from the extreme ends, then you will have a large charge separation, meaning you will have a large DC voltage develop, and they will be effective in charged based separation, as I shown in the previous slide. So our question is, can we develop a simple mask designed in such a way that where tribo charging will work? So our mask, what we developed after optimization, we choose these three materials, and cotton as a middle active layer, which is modified polypropylene layer, and the modification was done with the help with the ink made in our lab. This ink contains a graphene-based composites and then a nylon layer. So what we ask is that just by simple finger pressing, or even the simple movements as a jaw movement while we are wearing the mask, and if you talk, and this kind of sliding and releasing of layers, active layers, should able to develop charge in between. And what we found is that the modified masks are washable and also they are rechargeable. I'll show you the data. And then we found that the efficiency is also of the order of more than 95%. Then the next question is, like, once you designed a mask, how do we test it? What are the things to be tested to see the efficiency of a mask? One thing is sure, you need to show the particle permeation. So you need a particle count meter both in and out. So if these numbers with in and out will tell you how efficient this mask in filtering out the particles of the concerned size. And another thing is a humidity meter. And humidity, why it's important? Because if you can effectively stop water droplets, the modification is with a hydrophobic layer, you can effectively stop the water droplets going inside, and hence the droplet-based virus transfer can also be stopped. And then what is more important is that along with the efficiency of a mask, you should have a minimum pressure difference between in and out of the mask. If you have a large pressure difference, you will have breathing difficulty. So a mask with many layers may cause you a breathing difficulty. So a parameter called quality factor, a mask should have a good quality factor along with the filtration efficiency. So these are the sensors we have used in the mask testing device, and the concerned particles of size are sprayed using a simple sprayer. So this is a very simple schematic of the filtration measurement setup we have used. And I will show you the schematic and of the device, the actual device being developed. But let me show you the data from these things. So as I told you, what we are concerned is how many particles are being transported through the membrane with mask and without the mask. So if you count the particles of size of around more than 0.3 to 0.5 in this range of micrometer, what we found is that if no mask is used, almost 6,000 particles are being detected in the counter. With the commercial mask and our mask, you can see that the number of particles are really drastically went down. The efficiency calculation shows that, it has 95% or more efficient for particles of sizes around in the range 0.3 to 0.5 micrometer. This shows that this has the standard of N95 in terms of the filtration efficiency. and the mask works for the particles of submicrometer too. Now, the next question is about how long the charge can be retained in the membrane or in the mask. So in N95, one of the problems I told you is within a day or so, the charge will be completely drained off. So this is what we observe. N95 has the charge is completely gone in a day or maximum two. But with our mask, we found that the charge is retained even up to 5 days. And along with the charge retention, so this mask can be extended, use can be extended before recharging. But as I told you, another thing, if once it is completely discharged, can we recharge it or not? N95, you can do that to a certain extent, but the voltage generator will tell you how much charge is being generated. You can see that in our mask, it has a much larger voltage generation than N95. Yeah, just simple proceed. So this tells us that the mask is rechargeable, electrostatically rechargeable by simple finger press, and also the charge retention is also much higher than some of the commercial masks available. And hence, so this is the mask testing device being developed at the TIFR Hyderabad. This has the sensors like particle fluence and particle filtration efficiency sensors and humidity meters, and along with the pressure difference calculation to know the quality factor. And regarding the mask and the tester, we are collaborating with the local industry and to make them available in mass scale production or to reach the technology readiness level 0 to technology readiness level 9. Thank you for your kind attention, and the program and the research was fully supported by TIFR Hyderabad and Department of Atomic Energy, India. Thank you. I'm happy to address the questions now.
Thank you, TNN, for this talk. I'll just check if we have any questions. Can you please explain when would you expect to have this user-friendly, or can you let us know when would expect to have this user-friendly mass testing device developed?
Yeah, so the thing is that we have optimized all the sensors and also optimized the design. So we are working on the sensors what we have shown schematically here. So this will be ready very soon.
Okay. Thank you, Professor Nanan, for this talk. And with that, we come to the end of today's program. And thank you, everyone, for sharing your research stories. And we hope to listen to more such stories in future. At this moment, if you check our social media accounts, you're going to find that the link of the Science Media Center website has been posted. And before we go, there's one small announcement that I would like to make. Yeah, I was just hoping to share the screen. Yes. So on October 21st, we have Sawal Jawab, and this Sawal Jawab is going to be a session discussing this year's Nobel Prizes. We have 3 speakers, Krishnan Harshan, Parmeshwaran Ajit, and Sonal Nagarkar, discussing the Nobel Prizes. And it will be great if you all could join us virtually. The links are going to be up on our social media accounts. And with this, I thank all of you for joining in at today's program and hope to see you on the program on October 21st as well. Thank you, everyone.